This video is covering some material in Chapter 13. From now on, we're only going to be covering a portion of each chapter, and we'll do a portion of 5 different chapters over the next 5 days. So Chapter 13 is actually another review chapter, and that's because this is the sort of the introduction chapter to the section of the book about bioenergetics. So what bioenergetics is essentially just the study of how all these chemical compounds get converted in order to provide energy for our body. So just here's a quote from Lavoisier: respiration is a slow combustion of carbon and hydrogen. And that's really what it is. So you eat sugar, and over many different steps and many different reactions, it gets converted into CO2. You could have just burned this. You could set some sugar on fire and you'll get a lot of energy out of it because it will catch on fire and it will burn, right? You would get the same end products of CO2 and water. The respiration just does it in a much more slow, controlled manner because obviously we don't want flames in our body. So, but it's sort of the exact same thing where you get energy out of it. All right, so we are going to be talking about the basics of this in this term, and then in the second term of biochemistry, more detailed aspects, because that's a little bit more what it's about, but many people don't take the second term, and so this is sort of the highlights that are the most important to know about. So we're going to talk about some background information that's a little bit more important at this point since we're talking now about bioenergetics. So one thing is that we still have to obey the laws of thermodynamics. There's nothing special about life that it somehow gets to supersede these things, right? So we cannot create or destroy energy. It can just be converted into other forms. So you have energy in chemical bonds, you have energy in the form of heat, and you have maybe some other potential energy or kinetic energy or whatever, but you can't create it or destroy it. You have to just convert it into other forms. We are mostly going to be talking about changing different types of chemical energy. The second law of thermodynamics states that the universe tends toward increasing disorder, and this really is the basis for why some reactions happen and some do not. This leads us eventually to delta G, and tells us that reactions will only happen if they are thermodynamically favorable. So, and that has to do with the disorder of the entire universe. So remember here, we've been talking about delta G's a little bit here and there, but we're really going to it's going to be really important in this section. So the delta G standard, and remember this just means biochemically standard, is related to the K, the equilibrium constant, but delta G is also dependent on, the actual delta G is also dependent on the Q, the concentration of the reactants and the products. All right, so we know that delta G relies on the delta H of the reaction as well as the temperature and the entropy. Again, we're not going to worry too much about this. We're really just going to focus on the delta G. And so this is the actual delta G, but the delta G standard is related to K, right? So delta G standard is negative RT ln K. So this means reactions with Ks that are greater than 1, which would mean that at equilibrium there are more products than reactants, have negative delta Gs, and they're therefore favorable, and vice versa. So the actual delta G, though, so right, this is the delta G standard where all the reactions and products are all at 1 molar. This is really going to be very rare. Most of the time when you're doing a reaction, you do not already have 1 molar of both your reactants and products, especially the products. So the real delta G that's happening there is delta G standard plus RT ln Q. Q is just, right, the concentrations of the products raised to any coefficients of the reaction over the concentration of the reactants. So if you have a normal reaction you might set up in a lab where you just put the reactants in and you don't have any products, Q is essentially 0. So it's basically 0 divided by a really big number, right? So Q is essentially 0. You can't actually take the natural log of 0, but we could say it's like really, really tiny. If somehow you already only have products, Q would be essentially infinity. In practice, most of the time, Q is somewhere less than 1 because we don't have a lot of products. But in biochemical reactions, it's not quite as simple as you set something up in a test tube. So usually you actually do have some of the products around, but generally speaking, not so many that it's going to shift the reaction. So we are usually going to worry just about delta G standard and assume that it is relatively similar to delta G. There will be a couple of situations where this will be obviously not true, and I will point that out. So the other thing we know is that ATP hydrolysis releases energy, right? We take ATP, we cleave the third phosphate off, so we have ADP, and this is going to have a quite negative delta G standard. We'll actually see in a second that in this case, the real delta G is actually even more negative in most cases than the delta G standard. So assuming that this provides a lot of energy is a totally fair assumption. So remember, we're going to then couple this reaction to other things to make them favorable. So if we have, say, glucose plus phosphate goes to glucose 6-phosphate plus water. We've seen this reaction before. This hexokinase catalyzes this reaction, and we'll see it again in the next chapter. So this has a positive delta G, problematic. If we couple it to ATP hydrolysis, we should be able to make it favorable, because when you add two reactions together, we add the delta G's. So if instead we have glucose plus ATP goes to glucose 6-phosphate plus ADP, right, we can cancel out these things because they show up on both sides. And we just add these numbers together and we get a negative delta G, suggesting that this will happen. Again, we might have to worry about the really delta G standard versus delta G, but we're assuming this is going to be close enough. The K's are multiplicative. If you knew the K's, we more care about U delta G's usually, but K's are multiplicative, and you can realize this by actually just writing it out. So if you write out the K for this reaction, that's here. K for this reaction, that's here, and then we can actually, again, cancel out the things that show up on the top and the bottom. Should be the same things that canceled out up here. And we just get that the K is glucose 6-phosphate ADP over glucose times ATP, and that is the K of this reaction. So if we knew that the KQ of this reaction was some specific number and KQ for this one is some specific number, we could figure out the KQ for this reaction by just multiplying them together. So delta G's are I think more, people understand more why you add it together. Multiplying sometimes makes it tricky, but again, we will be focusing more on delta G's. So I said just a few minutes ago that the actual delta G of ATP hydrolysis is generally more negative than the delta G standard, and that's because there's a lot more ATP in the cell than either ADP, which shows up in the Q reaction, or AMP, which does not, but just also maybe worth mentioning that this is generally pretty low. So cells generally have much higher concentrations of ATP than they have of either of the other forms, right? So in a human red blood cell, 2.25 millimolar ATP, about 10 times less than that ADP, and another 10 times less than that AMP. So if we were to calculate this Q, it is generally going to be less than 1. Natural logs of things that are less than 1 are negative, and therefore this whole term will be negative and make the delta G even more negative. So this is why it's actually pretty reasonable to simply use the ATP hydrolysis delta G standard, because if anything, we'll be underestimating it. And while that's not the biggest of deals, you definitely wouldn't want to massively overestimate it, right? So this is sort of a unfortunate problem in that the more ATP you use up, the less efficient it gets. So if you start to get into a situation where maybe you have used up all your energy because you haven't had anything to eat or whatever, it also actually makes the ATP that you do have left work not as well, which is kind of unfortunate, but is reality. So when we talk about coupling these reactions, I think sometimes people just think that they're sort of like right next to each other and just happening next to each other, and they don't understand how that could possibly matter. But that's usually not what's happening. Usually it changes the entire mechanism. So we might write it like this, where the important reaction we care about is glutamate plus ammonia going to glutamine. Okay, and then we'll just put this ATP goes to ADP plus phosphate up here, sort of as a side reaction. However, it's not that these two things are happening simultaneously. It's that the ATP is part of a two-step reaction, where actually first we get phosphorylated on the side chain, and then the ammonia kicks off the phosphate. And so the it's not that this is wrong, it's just a little incomplete. All right, so essentially, if you see ATP is in a reaction like this, you can pretty much assume something on the organic molecule is going to get phosphorylated. Sometimes temporarily, right? Maybe this only one enzyme does this, so we don't really consider this to be a real intermediate that you could say isolate, but it is going to get phosphorylated. So ATP, we know ATP is used now to provide this energy, but where do we get it from? Okay, you do not eat ATP. You eat food, and then it is going to get somehow converted into ATP. And again, not even converted into, but the energy is. So we have a lot of energy from the food that we eat, and that breaking down that food is called catabolism. The energy from that is used to make ATP from ADP. So we don't make it from the food we eat. We just use that energy to take ADP and phosphate and put it together to make ATP. And then we use all that ATP to provide energy for these other reactions, various other things. Anabolism would be building up things. So if you want to make proteins or make amino acids, muscle contractions, active transport, we've seen both of these things before. So we need to use this energy. So essentially what ATP is a type of currency. So I think this analogy, we'll see it again in just a second, but this analogy works pretty well. When you're breaking down the food, right, say you have a big meal and later that day you're going to go running. It doesn't make sense to just store all the food literally maybe in your stomach or something, and then only when you go running break it down and use that energy directly. Okay, so instead what happens is we kind of break it down when you eat it or after you eat it, and then we store the energy in this form of ATP, and then whenever you do need it, we can use it there. We also have already talked about the fact we can store some of those molecules like glucose or fats for later use. So we absolutely can do that too. There's a lot of different ways to store energy, but for sure some of it is going to be converted just into ATP for a very short-term use. So what we're going to be focusing on in the next few chapters is this, how we take the energy from catabolism and use it to make ATP. We are not going to talk about anabolism in this section. We will get to that in the second term. So where does all this free energy come from? It comes from redox reactions. And we all know that redox reactions move electrons. They transfer electrons from one thing to the other, which changes the oxidation state. And in this case, in biochemistry, generally what it's going to change the oxidation state of is a carbon. So here we have 5 different variations of carbon. We have an alkane, an alcohol, an aldehyde or a ketone, a carboxylic acid, and then carbon dioxide. And these all have different oxidation states. I am not going to ask you to calculate the oxidation states. For biochem, we can essentially operate on a know just by looking how oxidized it is. We'll talk in just a second how we can determine that, but essentially the more oxygens it's got, the more oxidized it is. So that's actually a pretty useful way to do it. Why does that create energy? And an analogy maybe to think about is how a light bulb works, or a flashlight or something like that. And that's just that anytime we turn on some electricity, I mean everything is running my computer and everything, that's because we have electrons flowing through a circuit. So when electrons move, that creates energy that can be in the form of light or heat or something like that. We can also use that energy to make ATP. And it's going to be kind of a multi-step process before we get there. So I think that, if you think about that way, that redox reactions are going to allow us to use the energy that comes from moving the electrons. So redox reactions are famous for being able to be split into two. They have to be paired. You cannot just do an oxidation reaction. You have to do an oxidation and a reduction half reaction. That said, in biochem, we are often going to talk about oxidation or reduction, and the reason for that is we're only going to care about one of these two. They're both going to be happening, but the other one and a half of it is just going to be happening essentially to get the one that we want to happen. Okay, so keep that in mind, I guess. You can't have just an oxidation reaction, even though I'm absolutely going to talk about reactions and say that they are oxidations. So we can express these as half reactions where we just take the thing that is being oxidized, and remember that's the one that loses the electron. This is relatively easy to see when we have these nice inorganic metals that we can really see where the electrons are going, but it is true for biological ones as well. So this is a redox reaction, taking an aldehyde and hydroxyls, copper, and going to a carboxylic acid, copper oxide, and water. It is much less obvious how that's a redox reaction, but it is. Okay, and we can split it into essentially the organic one, so the aldehyde going to the carboxylic acid, and that also requires the OH and some water, and that's losing electrons. And then the copper reacting to form copper oxide, so this is copper 2, and this is going to be copper 1. It's kind of hidden in it's in a compound now, but so that took electrons to do it. All right, so this is a situation where we might really only care about this reaction, okay? We honestly don't care what's going on with the copper. What we want is to take an aldehyde and go to that carboxylic acid. So it's easy to call this a oxidation reaction while sort of ignoring the fact that there is also a reduction happening down here. So in many types of chemistry problems, we might actually want to calculate the oxidation state. However, in biochem, we can usually skip that step as long as we have a good sense because there's simply not as many elements involved. So all we really need to know is really about hydrogen, carbon, and sometimes nitrogen. But even we're not going to worry about nitrogen this term because we're not actually going to be talking about the oxidations of amino acids. So we're really only going to care about carbons that are bound to hydrogen or oxygen. So carbon is a little more electronegative than hydrogen. So when you have a carbon-hydrogen bond, we would consider that carbon owns all the electrons in the bond. Well, both the electrons in the bond. So this carbon, because it's in methane, actually owns all 8 electrons around it. All right, carbon came in with 4 electrons, so that would give it actually an oxidation state of -4 because oxidation state, oxidation number, -4 because it came in with 4 electrons are negative, it has an extra 4. This is the most negative oxidation state carbon can be in, is in methane. Now in something like an alkane, we have a carbon-carbon bond, and that's it's a perfectly shared covalent bond, so it doesn't make sense to assign it to one or the other, so we would say each carbon owns 1. So that has it has 7 and an oxidation state of -3. We consider something more oxidized if it has a higher oxidation state, so this is a little bit more oxidized. Even though we tend to think of these as being very similar things, it is actually slightly different. And on the way up here, at the other end of the scale, we have carbon that's bound just to oxygen, so carbon dioxide. And oxygen is more electronegative than carbon, and so in this case, all the electrons belong to oxygen, not carbon. Carbon owns no electrons. It came in with 4, it had to give up, so we would consider that to be a +4 oxidation state. So this is the most oxidized that it can be. So from the least oxidized, the most reduced, which is methane, to the most oxidized, which is carbon dioxide. And so we can also see anywhere in between. I want to, I guess, point on this one. Alkenes are more oxidized than alkanes because they have more bonds to carbon than they do to, to hydrogen instead. Alkynes are even more oxidized. And then we also start putting in alcohols, right, and oxygens, alcohols, formaldehydes, aldehydes, ketones, carboxylic acids. So, generally speaking, we're not going to see methane itself very much, but we will see alkanes and alkenes. You should definitely know how that works. We're also not going to see some of these special cases like formaldehyde or formic acid, or carbon monoxide. But definitely you should be able to look at it and say, okay, an aldehyde versus a carboxylic acid. Carboxylic acid is more oxidized, right? An aldehyde versus an alkene. Aldehyde's more oxidized. So I'm not going to ask you for these oxidation states or how many oxygens it owns, but you should be able to determine if a change happens in a carbon, you should be able to determine whether it got oxidized or reduced. And I would say generally you want to be able to do this pretty quickly and consistently. You don't want to have to sit here and think for a while about it. You want to look at that and go, oh yeah, it's an alkane, super reduced. Right? So when I say, so if we think about what this is, our food is and what we breathe out, our food starts generally as maybe alkanes or alcohols, maybe a little bit of carboxylic acids on those fatty acids, but the fatty acids also contain those really long carbon chains. Our sugars are generally going to have all those alcohols in them. So our food starts out pretty reduced, and we're going to breathe it out as carbon dioxide. So this is a consistent oxidation of the carbon. So I think I've kind of already described a lot of this, but you definitely need to be able to determine whether something's being oxidized or reduced, or neither. And so in Gen Chem, we would actually count up electrons. We'd look at whether they were lost or gained. That is generally not going to be the way you want to do it in biochem. What you really want to look at is, did the carbon gain an oxygen bond? So it doesn't have to always be a new oxygen atom. So if you go from an alcohol to a ketone, for instance, it gained a new bond to that oxygen. So that would still be considered gaining an oxygen. So that's going to be oxidation if it gained an O. Similarly, if it loses two H's, it's going to be an oxidation. And on the other hand, losing an O or gaining two H's is going to be a reduction, right? So one thing I want to point out is if we do both of these things, they cancel out. So if we do have something that loses an O and then also, if it loses an O and then it also loses two H's, say it loses a water, that is not going to be a redox reaction because it was neither oxidized or reduced. So one reaction that we were talking about in lab is the conversion of lactate to pyruvate from lactate dehydrogenase. And so you all are pretty familiar with this, and what we're doing here is this alcohol on lactate getting converted to a ketone. So we can look at this and go, all right, actually, usually you can do it either way. So you can look at this and go, this carbon, only one bond to oxygen, now two bonds to oxygen, that looks like oxidation. Most of the time, you're also going to lose two hydrogens. So we also could look at it and say, one hydrogen, two hydrogens, those are gone in the product, so we lost two hydrogens. This is not always the case. There are a few examples where, well, there's certainly going to be examples where we might lose or gain hydrogens with no oxygen involved at all. Conversion between alkanes and alkenes would do that. And there are going to be a few where we might affect the oxygens without the hydrogens. But in a lot of cases, we will actually do both of them. Again, make sure you're not doing one from this row and one from this row. Otherwise, that's not a redox reaction at all. All right, so this is an oxidation. If we went in this direction, it would be a reduction. So we've also seen in lab that you can't just get this to go by itself. You cannot just take lactate and lactate dehydrogenase and turn it into pyruvate. We need something else. And the reason we need something else is we need something to be reduced while that's happening. So really what that means is that we need something to transfer those electrons to if our main reaction we want to do is an oxidation. So those electrons can be transferred in one of 4 ways. Just directly onto something like an inorganic metal, so really just moving only the electron. That absolutely can happen, but is pretty rare. Or we can, along with the electrons, also move some protons or something like that, basically other parts of an atom. So we can just move an entire hydrogen atom. You may not think of that as moving an electron, but of course each hydrogen atom has an electron on it, so you're moving a proton too, but it's the moving of the electron that makes the difference. You can also move them as hydride ions. These are ions we don't think a lot of, but they're negatively charged hydrogens, so you take the hydrogen and add an extra electron, and that would be a hydride, and this is actually quite common in biochem. Or we can actually directly use oxygen. So say we could take this methyl group, actually take oxygen, and then turn it into an alcohol. This is also going to be quite rare. So really these two are going to be the more common ways to move hydrogen atoms or hydride ions. So as mentioned, breaking down food is overall an oxidation. We take, say, glucose, and we add oxygen that you breathe in, and eventually we breathe out carbon dioxide, and you either breathe out the water or you pee it out, sweat it out, something like that, or use it for other purposes, but we produce water. So this has a really negative delta G, really negative. Look at that, negative 2840 kilojoules per mole. That's very negative. That's a lot of energy. Okay, all of that energy comes from this oxidation that moves the electrons. So as we're doing this, we're going to be producing a lot of electrons. What we're going to do is we're going to use those electrons to make ATP. So essentially, by using these electrons to make ATP, we are using this energy that we can symbolize with delta G. So just like with the ATP, where we usually don't wait till we need it to make it, we don't generally want to wait until we need something else to be reduced in order to oxidize our glucose. Instead, what we're going to do is we're going to move the electrons onto electron carriers. This also makes the reactions be a lot more easily standardizable. So just always move electrons onto electron carriers, and then we can use the ATP to do any reduction reactions maybe we want to do later, or other electron carriers. So let's say we're going to take our molecule in the reduced form, convert it to molecule in the oxidized form. So we're going to start with an oxidized electron carrier and convert it to a reduced electron carrier. The most common one of these electron carriers is the NADH-NAD+ pair, the one that we've been using in lab. And there's also a variant of this, NADP+ and NADPH. So this is almost like we've taken 2 nucleotides and stuck their phosphates together, except this is not one of our normal nucleotides. This is a different base called nicotinamide, and that's where the N comes from. So everything's going to happen up here on this pink nicotinamide portion. The rest of it down here is nothing's going to change between it. So the one drawn over here is the oxidized form, NAD+. So one thing for sure also you just want to have cold is that NAD+ is the oxidized form and NADH is the reduced form. So what happens is here we have this aromatic group with the nitrogen that's positively charged. So when we have NAD+, and say we create 2 electrons and 2 hydrogen atoms from an oxidation half reaction. Most of this is going to end up on the NADH. So actually what's going to end up on the NADH is 2 electrons and 1 proton. So that's supposed to be H+. Okay, so 2 electrons, 1 proton is actually a hydride ion. But the other proton got taken off too, so we can't forget about it. And so it's just going to end up over here. That's fine, right? We got bases someplace that can deal with that. So having a proton is not a problem. We can't leave electrons floating around by themselves, but we can leave protons. So NADH is actually going to pick up the hydrogen here, or the hydride here, and so it's actually like this hydride. It's going to go on here. Electrons are going to move down, and then these electrons are going to move on to the nitrogen. So it makes this nitrogen not positively charged anymore, even though the hydride doesn't actually go there. It goes up here. All this is A side, B side is just saying if you can tell the difference between these hydrogens, right? There's the new one and there's the old one. There is now a sidedness to it, and each reaction that reacts with NADH or an NAD plus is going to put its preference like specifically on one or the other. We're not going to worry too much about that, but just kind of want to clarify why that was there. So the reaction that we always talk about is NAD plus 2. This just means hydrogen atoms. It does not imply that they were hydrogen atoms to start with. It means they came from somewhere, some oxidation or half reaction. Going to NAD plus H plus H plus. So actually, we don't usually write this. If you're writing one of those two reactions where maybe we have some oxidized or some reduced molecule here going to some oxidized molecule here, what you would have written is NAD plus going to NADH plus H plus, because this 2H is going to be part of that reduced molecule. So if you see this NAD plus going to NADH plus H plus, if you see that on a reaction, you can automatically know that what's happening here is an oxidation reaction. If you see the reverse, you can know that it's a reduction reaction, but we just won't be seeing very many reduction reactions this term. So the only difference between NADP is that there's a phosphate group here. So it's so far away from what's happening that essentially you can consider it, chemically speaking, like this to be, the same things happening as NAD+. That is a big enough difference, though, that most enzymes are going to use one or the other, and they are actually used in pretty different situations. We will really only see NAD+ in this term. We will see that next term. But the chemistry of the reduction is the same. So what we can do is we reduce this NAD+ while we oxidize this, and then at some other point, this NADH can go somewhere else and get oxidized back, and then we can just keep reusing it. Okay. So I've already mentioned this, but I just want to keep hammering it home because I think it's very confusing for a lot of people how we can have NAD+ that gets an H and then becomes NADH. So a lot of people, I think, we're so used to thinking of H's as being protons, like in an acid-base reaction, that we don't consider this, right? So that's how we transferred this hydride ion onto the NAD+, and that's why it was plus, and now it is neutral, and it has an extra H. And then we also have this H over here, right? So this is just an example of how this might happen, right? We have ethanol and NAD+ going to an aldehyde, and this is acetaldehyde and NADH. So oxidation half reaction, reduction half reaction. There's another carrier we'll see occasionally called there's actually two other ones, which are both flavonucleotides. The much more important one is flavin adenine dinucleotide, or FAD. And so FAD has, again, this looks sort of basically like it's a nucleotide here, but then we have attached something else. And FMN is just the part that doesn't have the dinucleotide on it. It doesn't have this portion, but again, everything happening is up here. So chemically speaking, they're similar, but we'll see FAD much more. So that's the name really that you should know of. What you should know about FMN basically is it's kind of a similar thing to FAD. So FAD is a little bit different. FAD picks up hydrogen atoms as opposed to the hydrides. And it picks these up one at a time, but it can pick up two of them. And so it can pick up one, so it's going to be this ring here, ring system here. It's going to pick up one, and it's going to put it on this nitrogen. And that makes an FAD radical. But usually it does not stop there. Usually then it picks up a second one, and then it goes over here, and that allows the electrons to kind of pair up eventually and make no radicals. So FADH2 is the fully reduced form. So the one that you should know about here, right, is taking FAD to FADH2. And again, this is sort of plus 2 H's, where we're not specifying where those H's come from, and usually we won't usually write that out. It'll be part of the reactant that the other reactant, the one that's getting oxidized. All right, so this one, we'll see it less frequently than NADH, but it's also pretty critically important. So that's all the background information, and then the rest of this term we'll be talking about breaking down macronutrients. So we have 3 macronutrients: proteins, carbs, and fats. We've already talked about those compounds. And for this term, what we're going to do is we're going to talk about breaking down carbohydrates and breaking down fats. We are not going to focus on proteins. Amino acids are not a major portion of the energy that you get. So when you eat protein in your diet, you're not using most of those amino acids for energy. You're going to use them maybe to build up your different proteins. You're going to use the amino acids, but you're not just going to break them down for energy unless you're eating a pretty unusual diet. Most of the energy we get comes from either carbohydrates or fats. Amino acids are also, practically speaking, more complicated because they have the nitrogen in them, and there's 20 of them. Whereas we can look pretty much at glucose and maybe a sort of representative fat, and we can be pretty, it's a lot more simple. So that's what we're going to talk about. And once we have finished how to break these guys down, then we'll have gotten sort of the basics of catabolism.